Today we are going to start another lecture that is regarding the peptides, protein, hormones in the endocrinology. First of all, we will discuss about what is the endocrine hormones are. The word hormone is derived from the Greek word hormo, meaning I excite or arouse. A hormone is a specialized chemical messenger that is a gland or tissue produces and secretes. So the hormone is a specialized messenger. Endocrine hormones affect the metabolic activities of the target cell or tissues in a specific way. As in the case of enzymes, hormones are not changed by the reaction they regulate. Only rarely does a hormone operate independently. More typically, one hormone influences depends on the balances for the another for the feedback. So one hormone is dependent on the another hormone. So the hormone is the chemical messenger, and it is derived from the Greek word that's meaning I excite or arouse. So biochemical classification, as we will discuss that proteins, amino acids, derivatives, and steroids. There are three classes main. Each class differ in a biosynthetic pathway, such as peptide and proteins are synthesized from the amino acids. Steroids are derived from the cholesterol, and amino acid hormones are the derivatives of the tyrosine. So peptide hormones, what are the peptide hormones? Peptide hormones are the made up of chain of amino acids that can range from just 3 to hundreds of amino acids. So peptides are linked to linked chain of the amino acids. Example are oxytocin and insulin. Their sequences are encoded in a DNA and can be modified by alternative splicing or post-transcriptional modification. These are the activities in the transcription, as in the biochemistry we are already discussed. They are packed in vesicles and hydrophilic. So they are hydrophilic and water lovers. So they are embedded in the vesicles, meaning that they are soluble in water. So the peptides are the hormones that are linked by the amino acid chain, and the hundred of chains are existed. And oxytocin is an example. Transcription modification may in these are present, and in the vesicles they are present. Due to their hydrophilicity, then they can only bind to the receptors on the membrane as traveling through the membrane in likely. So the hydrophilicity means they help in the traveling through the membrane unlikely. Some hormones can bind to the intracellular receptors. So some hormones are bound to the receptors, produce a mechanism called intracrine mechanism. Peptide and hormones, protein hormones, for example, most hormones are what are the protein hormones. Oxytocin was a peptide hormone, but now protein hormones synthesis, how it occurs. Most hormones are peptide protein in nature. The biosynthetic pathways are familiar with them from biochemistry. Like post-transcriptional modification is also occurring in the biosynthesis pathways, and the primary amino acid sequence of the peptide is directed by the specific messenger called ribonucleotide. Ribonucleotide in the messenger RNA, which has been transcribed from the gene to the for that hormone. So biochemistry processes occurs here, and the steps involved in the sciences are transcription. First of all, transcription occurs in the cytoplasm. Prepro hormone takes this activity, do this activity, essential of a prohormone, then packaging of the then secretion of the hormone occurs. Transcription, how transcription occurs? The transcription of the hormone from the gene from the DNA to messenger RNA. In the nucleus, the gene for the hormone is transcribed into a messenger RNA. Gene will transcribed into the messenger RNA. First of all, there is a central in the central dogma the DNA lies, but after that the messenger RNA forms by the help of transcription mode, as you already know about it. Generally, a single gene is responsible for directing the primary structure of the entire each peptide hormone. Because of the genes for almost all the peptide hormones have been cloned, recombinant DNA technology makes it possible to synthesize human peptide hormones. Okay, and now the sciences of the prohormones. The prohormones are transferred from to the cytoplasm and translated into the ribosomes to the first protein product called pro hormone. So the pre-pro hormone is the messenger RNA. First product is the pre-pro hormone. Then translation of the messenger RNA begins with a single peptide at the end terminus. So there is a terminus present. So the single peptide terminus, that is N terminus, at that point of translation occurs. Translation ceases, means stops. The signal of peptide attaches through the receptor on the endoplasmic reticulum via docking proteins, with the help of docking proteins. So the translation ceases means formation of the messenger RNA ceases, and translation then continues on the endoplasmic reticulum until the entire product is produced. So the product is pre-pro hormone. So instead of the pre-pro hormones, the signal how as proceeding pre-pro hormone sciences. The signal peptide is removed in the endoplasmic reticulum, converting the pre-pro hormone to the pro hormone. Pre-pro hormone means initial product, then the pro hormone is formed. The pro hormone The prohormone contains the complete hormonal sequence plus the peptide sequence, which will remove in the final step. So it will remove in the final product, but the prohormone formation takes some of the other peptide sequences in the prohormone are necessary for the proper folding of the hormones, so the formation of the intramolecular linkages. Sciences of the prohormone, how it occurs. The prohormone is transferred to the Golgi apparatus, where it is packaged into the membrane membrane bounded secretory vesicles. In the secretory vesicles, proteolytic enzymes cleave the peptide sequences from the prohormone to produce the final hormone. So it is occurred in the Golgi apparatus. So the packaging occurs, and then the pre-pro hormone converted into the pro hormone. So the final product will be found here in the hormone. Other functions of the Golgi apparatus include post-translation modification such as glycosylation and phosphorylation of the hormone. So these are the steps that are involved in the glycosylation and phosphorylation for the formation of the glucose and phosphate groups. In sciences of the final hormone, how it occurs. The final hormone is stored in the secretory vesicles until the endocrine cell is stimulated. When the stimulation trigger comes, so the secretory vesicles work, and final hormone will formed. These membrane bound secretory vesicles contain pro-hormone can be secreted from the from the cell by the exocytosis to produce to response on specific stimuli. Increases in calcium ion channel and CMP amino CAMP concentration in the cytoplasm. So these are the stimulus for the formation of the hormone from the hormone in the secretory vesicles. These hormones are contained as superfluous unnecessary amino acids and residues that were needed in the direct folding in the hormone molecules into the active configuration, but have no function. Once some hormone folds, specific endopeptide is in the cell cleaves in the prohormone just before it is released into the bloodstream. Generation in the mature hormone from the molecule. So this is the way how the hormone formed. Final mature peptide hormones then travel through the blood to all of the cells of the body, where they interact with a specific receptor on the surface of the target cell. So the final mature hormones are transferred to the target cells. For example, PTH, parathyroid hormone, is synthesized and stored in the vesicles in the chief cells of the parathyroid hormone gland. So these are stimulated by the calcium. These are ions with that of concentration, and the when senses of the parathyroid hormone gland detect low extracellular calcium contraction concentration, the secretory vesicles are translocated in the cell membrane, where they execute extrude PTH with into the blood by exocytosis. The other constituents of the vesicles, including the copeptides and cleavage enzymes, are extruded with the PTH. So that's why the that's how the PTH is secreted from the from the vesicle, and cleave out by the exocytosis. Examples of the synthesis of the these are the peptide hormone synthesis by the DNA to messenger, and transcription takes place in the nucleus and ribosomes. In the on the ribosomes, pro hormones releases, and the endocrine reticulum, these are proceeded to the pro hormone, and in Golgi apparatus, they are in the within the secretory vesicles of hormones are produced. These are the chain series how the hormones are formed from the central dogma via post transcriptional modification. Examples of the sciences of the insulin hormone: endogenous production of the insulin is regulated in the several steps. For example, what are the steps at the transcription from insulin gene in messenger RNA stability, at the end of the RNA translation, in the post translation modification. So the examples of the sciences of the synthesis of the insulin hormone: pre-pro insulin formation takes place first of all, then pro insulin formation, then mature insulin. So as we have already discussed about the above mentioned hormone, that is pre-pro hormone formation one, like in the secretory vesicles, how they formed, how they for example, we have discussed about PTH and how it is formed, and then transcriptional modification takes first, then stability, then translation, and then post transcriptional modification occurs. So prepro hormones, how is formed? Prepro hormone insulin consists of the two polypeptide chains, A and B chain, linked together by the disulfide peptide bonds in the it is however first synthesized as a single prepro insulin in a beta cells. Insulin contains 24 residual signals of peptide. It direct nascent polypeptide chain. So nascent means a newly formed polypeptide chain will occur in the rough and plasmic reticulum as ribosomes are also present there. And the signal peptide is cleaved into the polypeptide, is translocated into a lumen of the ribosome for forming a proinsulin. So proinsulin will form the pre-proinsulin with the help of beta chains and alpha chains. The pre-pro insulin will transformed into the proinsulin. So proinsulin in the rough endoplasmic reticulum, where ribosomes also occur, falls into the correct conformation, and three disulfide bonds are formed. 5 to 10 minutes after its assembly, endoplasmic reticulum proinsulin is transported into the translation trans-Golgi network, where immature granules are formed, which are immature, but after that proceeding mature insulin will also form, transport to the TGN, which may take place 30 minutes. So in mature insulin, how it is found? Mature insulin, proinsulin undergoes maturation after that into active insulin through the action of the cellular endopeptide disease known as PC1 and PC2, with the enzyme that is exoprotease carboxypeptidase that will aid as proinsulin to conversion to the mature insulin. The endopeptide disease cleave at the 2 point where fragment of the C peptide, and second phase is the alpha beta chain linked by the disulfide bonds. There are endopeptide disease aid the So the cleavage sites are each located after a pair of basic residues, 64 and 65 arginine, 31 arginine, and 32 arginine. These are the basic residues released from the released in the while the formation of the insulin hormone. After cleavage of the C-peptide, these two pairs pair residues are removed by the carboxypeptidase, and these chains are released that are proinsulin to the BCA chain that after releasing through the proinsulin hormone, hormone, the resulting mature insulin is packed inside the more mature granules, waiting for the metabolic signals such as leucine, arginine, glucose, and mannose, and vagus nerve stimulation to be exocytized from the cell into the circulation. So vagus nerve stimulation or exocytized means bulge out and release out and the mature insulin is formed. This how this insulin formed, the translation and transcription occur, then folding occurs, and vesicle is formed, and cleavage out and carboxyl is mature insulin is formed. That was all for today.